It's my great pleasure to introduce Dr. Alan Shuldiner, today's seminar speaker. So Dr. Shuldiner is the John L. Whitehurst Professor of Medicine at the University of Maryland, where he serves as the director of the Personalized and Genomic Medicine program. He has also, for the past 10 years, been a vice president at Regeneron, where he leads the Regeneron Genetics Center. And so we'll hear about some of that work. So Dr. Schuldiner's work focuses on the genetics of multiple age-related diseases, type 2 diabetes, which is quite close to my heart, obesity, osteoporosis, and cardiovascular disease. He's really well known for being one of the pioneers of studying founder populations, particularly the Old Order Amish, where his group made the first observation of a null mutation in ApoC3, which decreased triglycerides and conferred cardioprotection. And so that was in 2008 or so. Now, 15 years later, drugs that block ApoC3 are being introduced into the clinic. We're participating in some of those trials at UCSD. And I can say from personally treating these patients that what you used to be able to say to them is just like stop eating if they had very high triglycerides. And now you can actually give them a reasonable option. And so that's really fantastic. And so those are rare variants. And more recently, his group has also pioneered the use of common variants for predicting whether people are going to respond to drugs. So there's a particular blood thinner where you have to get the thinness just right, and it's called clopidogrel. And so some people respond differently based on their genetics. His group has worked on that. So Dr. Shuldiner, by way of background, trained at Lafayette College and then got his MD from Harvard Medical School, went to the NIH for training in endocrinology, and over his academic career has published hundreds of papers, trained dozens of MD and PhD students, and sits on many different committees and editorial boards. He asked me not to embarrass him, so I won't start enumerating those. But I'm very excited for his talk in translational genetics. So please join me in welcoming Dr. Shuldiner.
Well, thanks so much. I mean, it's really a pleasure to be here. And in 45 minutes or less, I'm going to try to tell you a little bit about what I've been up to over the last 30 years or so. Both with one foot in academia still, as well as now new gig, actually not so new, 10 years now in industry. And I have to say the best part of my job now is building bridges between academia and industry. There are just so many synergies that can be created with these collaborations. And I think a few examples you'll see today that will prove that out. So my disclosure, you've already heard that I have a foot in two doors. I work at Regeneron Pharmaceuticals at the Genetics Center and receive compensation for that position, and also University of Maryland. So what I'm going to do today is kind of focus on how human genetics is now being applied in the pharma and biotech industry to identify new therapeutic targets and the rationale for why this is being done. And I think, you know, the great insights at Regeneron and also other pharma and biotech companies has been that the time is right, because now our genetics tools are really powerful in the ability to apply human genetics to understanding human biology and ultimately new therapeutic targets. And what we hope to do is make this whole therapeutic development pipeline a lot more efficient. So now, without human genetics, more than 90% of molecules that enter into clinical trials actually fail. And they fail largely due to lack of efficacy or due to toxicity. And this is at a stage where hundreds of millions of dollars have been spent on these molecules, only to be put up on a shelf somewhere and ultimately move on to the next opportunity. And this has led the field to the idea that really the preclinical models that we use to identify new drug targets, mice, non-human primates, et cetera, are really poor predictors of clinical benefit in humans. And also recent data that has now shown and borne out multiple times now that compounds that are supported by human genetic evidence are substantially more likely to succeed in terms of efficacy and lack of toxicity. And I'm bulleting here, three, good examples of this. SGLT2 inhibitors are now blockbuster drugs for type 2 diabetes, and they also confer protection from kidney disorders. And the initial insights into SGLT2 was actually based on human genetics, rare families that had familial benign glucosuria. And this is what led then the pharma industry to identify SGLT2 as a potential target for diabetes. And the rest is history. What is this here? Okay. PCSK9 is often regarded as the poster child for human genetics. I think most of you know that story. Rare families that have hypercholesterolemia and other families that have hypocholesterolemia with either gain of function or loss of function mutations in PCSK9, and now PCSK9 inhibitor monoclonal antibodies are FDA approved for the treatment of hypercholesterolemia. ApoC3, Amit already mentioned to you, again, a human genetics story that then led to therapeutic development, and now multiple others that I'll provide some examples of. So this has led the pharma industry, in particular Regeneron, about 10 years ago to found the Regeneron Genetics Center, in which we're now really full throttle into human genetics. And in fact, at Regeneron, unless there's human genetic evidence for a particular target, we don't pursue it. So the whole drug discovery paradigm has been turned on its head. Rather than starting with preclinical models, we start with humans and human genetics. And we use human genetics for target discovery to identify new targets and pathways. You can also use human genetics to find new indications for therapeutic targets through so-called PheWAS approaches. You can use human genetics to actually de-risk a particular drug target by confirming lack of on-target adverse side effects in drug target loss of function carriers. And of course, you can use human genetics for the traditional pharmacogenomics to essentially stratify patient populations by those who may be more likely to respond or not, or have adverse side effects or not. And only after the human genetics is done do we then begin to try to understand underlying biology. And that's when we go to the preclinical models. We use mouse genetics, we use in vitro systems, iPSCs, organoids, even human callback studies often to really drill down on basic understandings of what that drug target does and how it does what it does. So that's the paradigm. And now, about 10 years ago, the Regeneron Genetics Center essentially started, and we built a comprehensive genome center within the R&D program at Regeneron. We have an automated biobank that can store millions of samples, literally unlimited capacity, because when one fills, we just buy another and we fill that one and go beyond that. We also have developed high throughput automated sequencing and genotyping, and also everything we do in terms of data storage, data management, data analysis is done in the cloud, so almost unlimited numbers of electrons to perform the analyses that we need to do. To date, we've now sequenced over 2 million exomes from research participants. We're sequencing now at the rate of over 500,000 subjects per year, and I don't think there's any end in sight. And our recent focus has been on diversity to begin to better understand the genetic architecture, not just of European ancestry, Homo sapiens, but of allelic architecture of populations from around the world. And in fact, that's what attracted me to join Regeneron. And I'll tell you a little bit about what my group in particular has been doing in the founder and special populations space. So 2 million exomes, where do we find these DNA samples? And the answer is that we find these DNA samples through collaboration with academic collaborators and healthcare systems throughout the world. And we now have more than 120, I think it's maybe more than 130 or 40 now, collaborations in 23 countries. And we continue to grow the portfolio of global populations for genetics studies. And we're very opportunistic and even agnostic, I would say. We just want to apply human genomics to understand human biology, agnostic to disease or phenotype. And we need to be opportunistic. And the 4 buckets of collaborations that we have each have their own angle in terms of opportunities for novel gene discovery. We're very interested in general populations, particularly those that are linked to electronic health record data. So essentially, we can sequence DNA and have at our disposal the entire genome, and also through electronic health records, essentially access to the entire clinical phenome to do what we call all-by-all analyses and identify new hits for clinical phenotypes. We're also very interested still in family-based studies, probands and families with rare undiagnosed diseases. Often, just need to sequence one or a few individuals, and you can come up with new genetic hits, new biology. And often, although these are associated with or causative of rare diseases, there's often biology that translates into the more common disorders that affect and cause morbidity in populations. As you hear more, I'm really interested in founder populations, and I've done a lot of work over 30 years in academia with the Old Order Amish in Lancaster, Pennsylvania. There are thousands of founder populations all over the world. They're on hilltops, they're in valleys, they're on islands, they're religious isolates among us. And just as one founder population is an experiment of nature, and each is its own independent experiment of nature with opportunities for novel gene discovery. And I'll tell you a little bit more about that. And then there are some diseases where you can still recruit 1 million people and still have very few cases. And so we're very interested in working with academic collaborators who have spent their whole careers gathering individuals with a particular disease of interest in case control studies. So those are essentially the 4 buckets that we opportunistically use. We're phenotype agnostic, as I've mentioned. We work in all areas. Some, a little bit more targeted than others that I'm showing you on this slide: cardiovascular disease, non-alcoholic fatty liver disease associated with obesity, immune diseases, infectious diseases, cancer, eye, hearing loss, and others. And in that we work with academic collaborators from all over the world, we need to respect the currency of the academic community. And we provide DNA sequencing. We provide that data back to our collaborators. They utilize that data for their own internal research purposes, as we do at Regeneron. And it's fully collaborative in that we work together and we publish together. And I have to tell you, I've published more articles in higher profile journals as part of the Regeneron Genetics Center than I ever have in the 30 years that I've worked in academia. Examples shown on this slide. And it's been really proud that we've been able to enable scientists to do amazing science that they ordinarily would not have been able to do. Regeneron led a pharma consortium to essentially provide the funding to sequence all 450,000 samples from the UK Biobank. That data is publicly available, and there have been hundreds, if not thousands, of publications in the last few years that have utilized that data. And just recently, actually now in revision, but you can find it on bioarchives, we've actually now provided the allele frequencies that we've gotten from exome sequencing of nearly 1 million individuals that have been sequenced at Regeneron. And that data also is publicly available in a browser that anyone can glean. So how does this work? Let me give you an example or two. This work was published now about 3 years ago, 2.5 years ago, and it's the identification of a new gene through human genetics that we think may be a very effective target for obesity. The story starts with the sequencing of 640,000 individuals, and then performing essentially genome-wide association analysis for body mass index in this case. You can see in B that the discovery cohorts included, as I mentioned, about 640,000 individuals, and we found associations of loss of function variants in a gene called GPR75 that were associated with actually protection from obesity. You can see here the cohorts that went into this, the fact that these individuals had lower body mass index. You can see that these variants in GPR75 are very rare. Sequencing 640,000 individuals, there are only 228 carriers of loss of function variants in this gene. So this is human genetics at unprecedented scale to be able to find enough individuals with these rare and ultra rare variants that ultimately associated with a phenotype of interest. Of course, one wants to replicate that data, and you can see that in B below that we were able to replicate that data in an additional almost 90,000 individuals. And you can see here sort of the effect sizes, and these are all the hits with p-values below the threshold for genome-wide significance. You can see that in this analysis, we were able to identify most of the common variants that are associated with body mass index, each with relatively small effect. This is the so-called GWAS hits that we've already known about. And then when you get into the rare variant sector here, you can see we're finding some known variants that are rare variants that are associated with increased body mass index. And you can see here a few that are also associated with decreased body mass index. GPR75 is sort of the outlier in terms of rareness and also effect size, in that this looks like a difference of up to, well, these are standard deviation units. Yeah, here we are, up to almost 2 kilograms per meter squared in carriers of these loss of function variants. This is what this kind of looks like. You can see that across the gene, there are loss of function variants that we found in these 640,000 individuals. A lot of them are singletons, just present in one individual. But when you add them all up and you do these gene burden tests, you find those associations with decreased body mass index. You can see here that loss of function variants, those 228 individuals, there was a lot more relative to non-carriers that were of healthy weight, and of course, reciprocally fewer that were obese and severely obese. On the other side of the coin, the well-known mutations in MC4R that cause obesity, you can see that we found essentially the opposite. So a nice proof of principle. And then, of course, once you know the gene, you want to know what it's doing. You want to replicate in preclinical models. We went ahead and made mouse knockouts of GPR75. You can see that in A here, in response to a high-fat diet, those that were knockouts and even the heterozygotes for GPR75 gained less weight than wild type. And you can see also that metabolic complications of obesity were also ameliorated. You can see fasting glucose here in terms of the high-fat fed individuals, or mice, I should say. The knockout mice had lower fasting glucose. They had better glucose tolerance during oral glucose tolerance test, and also lower insulin levels, suggesting decreased insulin resistance. So a nice proof of principle that GPR75 could be a new target for protection from obesity or from treatment of obesity. GPR75 was not on anyone's map before these findings. In fact, very little is known about GPR75, but now we have the motivation to really better understand its biology. Potential ligand, so it's an orphan GPCR. Ligands that thought to maybe bind to GPR75 included some of the pro-inflammatory chemokines and eicosanoids. It is expressed in the brain, so we think there may be a central mechanism to the protection from obesity. But still the mechanism is still being worked out. But again, obvious that this could be a potential new target for the treatment of obesity. So that's one example of kind of how this works. in the population scale of hundreds of thousands of sequenced individuals. But maybe there are more efficient ways to do this, and this is where the founder populations come in, and where I spent the better part of my career working on. For those students who don't know the nitty-gritty of why founder populations are wonderful for novel gene discovery, this is a quick slide that introduces you to some of the concepts, population genetics concepts. For one thing, founder populations have a lot more genetic homogeneity than general populations, because the gene pool of that entire founder population came from a small number of individuals, the original founders of the population. And then if that population remains isolated from the rest through generations, there's the opportunity for genetic drift. Rare alleles that might have gotten into the population through the founders, even one founder, through multiple generations, could drift and increase in frequency, making that variant easier to find. And if it's associated with a trait of interest, of course, easier to find that association. Some of these founder populations, in that they marry among themselves, there's consanguinity. And often these populations also have large families. And this then provides the opportunity to not only find drifted variants that increase in frequency, but to find them in their homozygous state. We have examples of that too that I'll show you in some slides to come. And then finally, some of the confounding influences of finding genes, particularly for complex diseases in which there are gene-environment interactions, may be a little bit cleaner in founder populations, in that these founder populations tend to practice a more homogeneous lifestyle than general populations. And finally, in terms of recruitment, they tend to be geographically localized. So if you gain the trust of the community and their partners in research, you can actually recruit many of them within a relatively small locale. So that's why founder populations are so interesting for genetic discovery. As I mentioned, I've been working now for over 30 years with the older Amish in Lancaster, Pennsylvania. They have all the characteristics that I mentioned on the prior slide. They were founded by about 200 to 600 or so individuals who migrated from Switzerland about 15 generations ago. Sorry about that. Okay. And their current size is about 40,000. So the gene pool of these 40,000 essentially is the same as the 200 to 600 individuals who founded the population. And these genes have been shuffled and reshuffled through 15 generations, essentially, to the current gene pool today in Lancaster, PA. They've remained isolated, so very few new genes. When I say genes, I mean genetic variants in genes have come in to the population. And they're essentially a religious and a cultural isolate. Still, they shun modern technologies. They don't use or have any electricity in their homes. They use horse and buggy as their main mode of transportation, and that keeps them as a cultural isolate. Very large sibships. Average sibship size in the Amish is about 7. We have some sibships in which we have 18 and even 19 siblings from the same 2 parents. And there is this consanguinity in that the average relatedness is first cousins once removed, or another way of saying it is sharing one grandparent. Every Amish related to each other in those to that degree. And as I've mentioned, there's this opportunity for genetic drift and the characteristics of ultimately finding new genes. And maybe for myself as a physician scientist who at the end of the day, enjoys making these basic discoveries, once you've made these discoveries in these founder populations, in many ways, it's easier to actually influence in positive ways the health of the community, because you can actually catalog all of the possible pathogenic mutations that can ever occur in that population, and then utilize that information for population genomic health. And that's what the Amish Research Clinic has been doing now, and some really dramatic reductions in diseases in the Amish through essentially human genetics. Don't have time to go through 30 years of discoveries that we've made in the Amish. I'll just mention a couple. You've heard about ApoC3. It turns out through genetic drift, 1 in 25 Amish carry this null mutation ApoC3. They have low triglyceride levels. They're protected from heart disease. And this has now led to these the therapeutic development of ApoC3 inhibitors. From population genomics side, we found that 1 out of every 8 Amish has familial hypercholesterolemia, LDL levels that are really high, caused by this previously known R3527Q mutation in ApoC3. This was already known in the general population, but is very rare in the general population. But through genetic drift, 12% of Amish have familial hypercholesterolemia. Of course, we're now able to utilize that information to find children that carry the mutation, probably 20, 30, 40, 50 years before they would ever get a blood test, and be able to intervene early to prevent atherosclerosis in those children to be adults. We've found a new form of familial lipodystrophy due to mutations in LIPE, which encodes hormone-sensitive lipase. A lot of really interesting biology behind that, still working on some of that work. As was mentioned, we did a Plavix or a clopidogrel pharmacogenetics study in the Amish, and we're among the first to identify common variants in a cytochrome P450, CYP2C19, that predicts response to clopidogrel. And now cardiac catheterization labs around the world are performing genetic testing in their patients that get stents in order to prescribe in a much more precise way antiplatelet agents in their patients. And now, and of course, all of those findings were more or less fortuitous through candidate gene sequencing or common variant genome-wide genotyping. In recent years, through collaboration with the Regeneron Genetics Center, for the first time, genetically speaking, we're playing with a full deck. We've done whole exome sequencing and genome-wide genotyping and imputation in over 10,000 Amish individuals. And now with that data set coming up with all sorts of new and interesting findings. For one thing, we do have a catalog of 2,000 pathogenic variants that can be genotyped for, I won't say pennies, but for low dollars. And essentially now for population screening of essentially all the genetic diseases that the Amish could ever have. De novo mutations aside, but and that's being used now by the Clinic for Special Children in Lancaster for early diagnosis of all of these diseases. We found that 1 out of 45 Amish actually have a pathogenic mutation in a gene called KCNQ1 that causes long QT syndrome and increased risk of sudden death. That's one of those 2,000 variants that's now on the panel. And I'll tell you a little bit more about now a really interesting discovery in a gene called B4GALT1, again, that was never on the radar for being a potential target for hypercholesterolemia and protection from heart disease. So this story starts with the exome sequencing of a little under 7,000 Amish individuals. These were individuals that I spent 30 years recruiting at the University of Maryland. And we went ahead and did a exome-wide association study for LDL cholesterol. And you can see the XWAS, as we call it. And you can see that we have a hit here that was associated with lower LDL cholesterol, highly significant. It was also associated with lower fibrinogen levels. And it turns out that this hit on chromosome 9 wasn't present in any of the huge genome-wide studies that have been done in outbred general populations, most of them dominated by European ancestry. And the reason that it wasn't picked up in any of these very large studies is that this is a variant that is markedly enriched, in fact, almost private to the Amish. It's a single base change that predicts the amino acid change of asparagine to serine at position 352 of a gene called B4GALT1. The allele frequency is about 6% in the Amish, as I'll show you, almost absent in other populations. So what does B4GALT1 do? It's actually a galactosyl transferase. Who would have ever imagined that I would have any interest in glycoproteins and glycosylation. But the genetics have led us to this finding. And essentially, this is the galactosyltransferase that actually adds galactose to the carbohydrate moieties of glycoproteins. And that happens in the ER and in the Golgi, as you can see on the slide. This particular missense mutation occurs very close to the active site of B4-GALT1, which is a manganese-dependent enzyme. And we went ahead and expressed this missense variant in vitro. And on the lower part of the slide, you can see that this single amino acid change results in about a 50% decrease in enzymatic activity of the protein. And if you look in the serum of individuals that carry this variant, in fact, you can find that these individuals have less galactose and sialic acid on proteins, glycoproteins. So you can see total glycoproteins. You can see fibrinogen has less galactose and sialic acid, ApoB, also IgG, others as well. We went ahead and made knock-in mice for the serine variant. And indeed, these knock-in mice have lower LDL cholesterol levels, and they also have lower fibrinogen levels. And then we went ahead to look a little bit more closely as to what was the organ specifically that was involved here. We went ahead and performed CRISPR knockdown of B4-GALT1 in liver of mice. And sure enough, with about a 50% decrease in B4-GALT expression in the liver, you can see we were able to recapitulate the lower LDL cholesterol levels. So much of what's happening here is happening in the liver. And in order to try to figure out whether these lower LDL cholesterol levels and lower fibrinogen levels had any clinical consequences whatsoever, we had to go ahead to our very large populations and found that in 485,000 individuals, non-Amish individuals sequenced, we were able to find this variant in about 440 individuals. And sure enough, these individuals are protected from coronary artery disease with an odds ratio of about 0.6. So a completely new discovery that would have never been possible without this experiment of nature, founder population, genetic drift, and fortunate enough for us to have measured LDL cholesterol level in order to make the connections. And this has now introduced the idea that, maybe glycosylation is a pathway that we should be thinking more about as a non-traditional risk factor for cardiovascular disease and ways potentially to intervene with glycosylation in ways that might protect from heart disease. There may also be implications for immune disease and inflammation, and we're just now beginning to understand and study this in more detail. Okay, so I have two more quick examples, and I'll try to go a little bit more quickly because I'd love some time for Q&A. My group is also very interested in expanding genetic diversity and understanding allelic architecture across the planet, and also the relationship of allelic architecture to the increased prevalence of certain diseases that occur in various populations around the world. And as an endocrinologist, type 2 diabetes is still an area of interest for me, especially in this era of hyperalimentation and obesity, and now non-alcoholic fatty liver disease. It's a leading cause of liver failure. And it turns out that type 2 diabetes actually varies quite a lot in terms of its prevalence across various populations. South Asian ancestry individuals tend to have about a 4-fold higher increased risk of type 2 diabetes than European ancestry populations. Even at leaner BMIs, they're more susceptible to the metabolic complications of obesity. So this has led to the idea that maybe if we study type 2 diabetes in some of these populations, we might find new things. South Asians, as you know, comprise about 1/4 of the world's population, but in terms of representation in sequence data, they represent less than 2% of participants that have been sequenced in published studies. And despite the fact that very large GWAS studies have been done for type 2 diabetes, more than 611 loci that have been identified, common variants with each with small effect on risk for type 2 diabetes, very little has been done in South Asian populations. So we went ahead to go ahead and find collaborators who had access to South Asian DNA samples and phenotype data. We have a collaboration with John Dinesh at the University of Cambridge. He leads the BELIEVE study, in which he has recruited over 50,000 Bangladeshi in Bangladesh. And we went and did an exome-wide analysis of type 2 diabetes in these 50,000+ Bangladeshi. You can see that we're picking up a lot of the major hits that are already known, but you can see one hit here on chromosome 20 that is actually protective of type 2 diabetes. The triangle is upside down. And in fact, this is a new story that, again, could only have been discovered in South Asian populations. So it turns out that this is a variant in the HNF4 alpha gene. Single base change predicts a proline to serine change at position 437. You can see that the allele frequency in South Asians is about 1.2%. In the 45 or so thousand individuals, there are a bit over 1,000 heterozygotes, 14 homozygotes in Bangladesh. And again, they protect from people that have this serine allele are protected from type 2 diabetes. About a 50% reduction in risk. And as you can see, this is NOMAD data, but I can show you other data as well. This particular missense variant exists pretty much only in South Asia. You can see other European populations, African, Ashkenazi, et cetera. The frequency of this variant is pretty much zero. But it is about 0.5% in South Asians from NOMAD. As I've mentioned, particularly within Bangladesh, it's over twofold higher frequency than that, about 1.2%. And supporting data here shows that individuals that have this variant, not only are they less likely to develop type 2 diabetes, but if they do develop type 2 diabetes, that onset tends to be delayed by about 4 years. So the average age of onset of diabetes in carriers who are unfortunate enough to get diabetes is almost 50 years old, whereas those non-carriers, they tend to get the diabetes on average at around age 46. And also we have some preliminary data suggesting nominally that this variant may also protect from chronic kidney disease, something that we need to investigate in further detail. So HNF4 alpha has an interesting story. Don't have time to go through all of these bullets right now, but HNF4 alpha is known to have loss of function variants that are pretty rare that cause a form of diabetes called maturity onset diabetes of the young. It's an autosomal dominant disorder. It's called MODY1. And so we know that this gene was on the map, but on the other side of the map in terms of loss of function variants, increasing risk for diabetes. To our knowledge, this is the first mutation in HNF4 alpha that actually protects from diabetes. And we're now in the lab trying to nail down particularly what's going on here. But it turns out that this serine, Pro437 serine variant actually occurs in a highly conserved proline-rich C-terminal repression region of HNF4 alpha protein. And it suggests maybe that this particular variant actually results in a gain of function, i.e. loss of repression. It's a transcription factor and is very pleiotropic in terms of the large number of genes that it affects transcription for. And we're now in the process of figuring out specifically what genes this serine variant turns on at higher levels than wild type, which then may provide us some additional downstream targets that may be more effective and more specific. And as I said, HNF4 alpha has been studied for a while because of its relationship to maturity onset diabetes of the young. There are mouse models not produced or studied by Regeneron, but in the literature that support this story. Overexpression of HNF4 alpha in mice actually shows protection against high-fat cholesterol fructose diet, hepatosteatosis, inflammation, and fibrosis. And also overexpression of HNF4 alpha in liver of mice with small activating RNAs actually also show significant improvement in diet-induced glucose and lipid metabolism. So really a suggestion that potentially in humans, there might be the opportunity to activate HNF4 alpha or something downstream of HNF4 alpha that could protect or treat diabetes. The last story I'm going to tell you is a really cool one, a collaboration we have with Dinesh Salehin. He was formerly at the University of Pennsylvania, more recently at Columbia University. He leads the Pakistan Genome Resource. And Pakistan is really a unique opportunity to identify human knockouts. So we love mice and we love doing genetic acrobatics in mice to make mouse knockouts. Some of what we learn in the mouse knockouts do apply to humans, but a lot doesn't. And here we now have the opportunity for the first time using high throughput human genetics to identify human knockouts, and then through callback studies and the like, really understand what the consequences of knockout of a gene is in humans. So Pakistan is a large country, very diverse, many founders, subpopulations within Pakistan. Extreme consanguinity, more than 50% of marriages in Pakistan are in first cousins or even potentially closer. And as a result, there's a much higher frequency of homozygosity across the genome, but in particular for variants that result in a loss of function of a particular gene. So we're in the process now of sequencing hundreds of thousands of samples from Pakistan. So far in 80,000 exomes that we've sequenced, we have 6,400 genes of what, 25,000 or so, about a quarter of the genes in the genome. We have identified at least one human that is homozygous for a loss of function variant, essentially a human knockout. And as we sequence more and more, we'll identify more genes of human knockouts. Of course, there'll be some that are incompatible with life, and we won't find those, obviously. But this is an amazing project, the Human Knockout Project, in which we identify these individuals and then can call them back for deeper phenotyping. So the quick story I'll tell you so far is one of stroke. Again, in South Asians, particularly Pakistan, there's a much higher prevalence of stroke than in other parts of the world, as you can see here. And in particular, one of the rarer forms of stroke, hemorrhagic stroke, is one that leads the list in Pakistan. So not only a higher frequency or prevalence of stroke, but higher frequency of hemorrhagic stroke. Again, in collaboration with Dinesh Salehi and the Pakistan Genome Resource, we sequenced over 60,000, now as I said, 80,000 Pakistani. We had about 30,000 in what we call a discovery cohort and 30,000 in a follow-up cohort, and several thousand cases of stroke and controls that did not have a stroke. And then we followed that up with UK Biobank data, as I mentioned before, not only to look at stroke, but many in the UK Biobank have had MRIs of their brain, and we can actually then begin to dig more deeply into imaging data of individuals that have particular variants of interest. So this is what the exome-wide association study looks like for stroke in about 32,000 individuals from Pakistan. We had 5,000 stroke cases and 27,000+ controls. You can see we have one hit that achieved genome-wide significance, and it turns out that this is a single base change in a gene called Notch3, predicts the amino acid change of arginine at position 1231 to cysteine. And you can see the frequency in Pakistan is close to 6%. And it increases risk of stroke by about twofold of all stroke. You'll see hemorrhagic stroke, it's even a higher risk. And again, you couldn't make this discovery in European samples because this variant is much less common in European ancestry individuals. You can see that in Pakistan and South Asian populations, 1 in 94 individuals actually carries this missense variant and is at increased risk of a stroke, compared to European populations where it's closer to 1 in 2,000. So a highly enriched variant causing this missense mutation that increases risk for stroke in essentially a quarter of the world's population. We did some additional genetics of other groups in South Asia and the Middle East, and this variant actually is enriched all across the Middle East and South Asia as well. So this is kind of what the data looks like. You can see that we have the discovery cohort that I've already shown you, a replication cohort, and ultimately a highly significant association risk of about 2.5-fold for stroke for this missense variant. You can see that on the lower part of the slide, when you look at hemorrhagic stroke, you can see the risk is about 3-fold 3 to 4-fold for subcortical hemorrhage and intracerebral hemorrhage. Again, this is the form of stroke that's particularly enriched in South Asians, and now we think we know why. And then when we went to the UK Biobank, we were able to find some South Asians in UK Biobank that had MRI data and also had the variant. And these individuals have abnormalities in their MRIs, their brain MRIs, alterations in white matter hyperintensity. This is imaging data in which you can literally take each pixel and look for association of intensity of that pixel in carriers of a particular variant versus non-carriers. And you can see that in particular areas of the brain here, cortical white matter, periventricular white matter, internal and external capsules, you see alterations in white matter hyperintensity in ARG1231 cis carriers. So Notch3 is an interesting story. It's essentially a gene that encodes a cell surface receptor that's very important in vascular development and also maintenance of normal vasculature. And mutations in Notch3 have been known to cause a very rare form of stroke called CADASIL, cerebral autosomal dominant arteriopathy with subcortical infarcts and leukodystrophy. And these mutations are very specific in that they alter a cysteine within the EGFR repeat domain of the protein, which is at the amino terminus. And if you alter a cysteine, you have an odd number of cysteines, and that is thought to lead to aggregation of Notch3 in the extravascular space, so-called granular osmophilic material, which is pathognomonic of CADASIL. And that's what is thought then to cause the hemorrhagic stroke, the breakdown of the smooth muscle in the vasculature and ultimately hemorrhage. So the insight here is that actually CADASIL is not a rare disease in South Asians. And there's a milder form of CADASIL that occurs in 1 in 92 South Asians that puts them at increased risk for stroke, in particular hemorrhagic stroke. Classical CADASIL is caused by cysteine altering mutations in the first 6 or so EGFR repeats. This particular one happens in the 31st EGFR repeat, and that's why we think that it's actually looks like a milder form of CADASIL, one that actually is pretty much indistinguishable clinically from typical stroke or hemorrhagic stroke. And if you do the population genetics, the epidemiology, as I mentioned, this variant is present in about 1% of South Asian. It incurs a 3-fold increased risk of hemorrhagic stroke. It's attributable to approximately 1% of all strokes in a quarter of the world's population, and about 5% of hemorrhagic strokes in South Asians. So not a trivial disease burden for this particular population. I mentioned that we also are very interested in applying human genetics for pharmacogenomics to better understand variation in drug response, and just showing you this one slide that's already published in which we looked at polygenic risk scores and responsiveness to PCSK9 inhibitors. You can see that those who have a high polygenic risk scores are actually individuals who are more likely to see benefit of PCSK9 inhibitors. Again, something that could potentially be used clinically to target populations that are more likely to benefit from drugs. So that's my story. I could go on, but just to summarize for you, I think I've shown you how now new targets are being fueled by human genetics in the biotech and pharma industry. Large-scale DNA sequencing of what I call strategic populations is kind of the approach that we've been using. Showed you the examples of GPR75, HNF4 alpha, and Notch3. I told you a little bit about founder populations, still something that's of great interest to me. And as you see, we're sequencing founder populations all over the world. And overall, just the fact that none of this would be possible without these partnerships between industry and academia. And we hope to continue to bring on new collaborations that will have real effects, not only for target discovery and new therapeutic development, but also for the implementation of precision medicine in these populations. So thanks for your attention. A huge number of individuals who have been involved with this research. The RGC now has about 130 full-time scientists. And of course, our collaborators, University of Cambridge, Pakistan Genome Resource, and my colleagues at the University of Maryland studies in the Amish. Thanks for your attention. So just by way of one more sales pitch, Regeneron has a terrific internship and co-op program. So for any students, high school, undergrad, graduate student, you get 8 to 10 weeks to spend during the summer. I encourage you to look into our internship and co-op program. You can actually get your hands on all the genetic data that I talked about and do various kinds of analyses. For those of you who are getting your PhD degrees, also Regeneron has an amazing postdoctoral fellowship program that I'd be happy to talk to any of you about, or you can check out on the web. Thanks.